Welcome to this lecture on animal biotechnology. Today, we are going to explore one of the most fascinating and life-saving applications of modern science, recombinant DNA in medicines. Imagine if we could turn microscopic bacteria into tiny living factories that pump out exact copies of vital human hormones. Well, we do not have to imagine it because this technology exists today and keeps millions of people alive. Let us dive in and see how it works. To begin our journey, we must first understand what recombinant DNA technology actually does. Why does this matter? Because before this, we had no way to create complex human proteins outside the human body. By taking a specific human gene, the blueprint for a medicine, and splicing it into a bacterial plasmid, we create recombinant DNA. We then place this into a host organism, usually a harmless strain of E. coli. Because the genetic code is universal across all life on Earth, the bacterium reads the human DNA just like its own. It becomes a living factory, mass-producing pure, human-identical proteins that we can harvest for patients. Now that we understand what the technology is, let us look at why it was so desperately needed. Before recombinant DNA, patients lacking vital hormones faced a grim reality. They had to rely on medicines extracted from the pancreases of slaughtered pigs and cows, or even human cadavers. This was incredibly problematic. Animal insulin is slightly different from human insulin, which caused severe allergic reactions in many diabetics. Furthermore, harvesting from cadavers was not only expensive and inefficient, but it carried massive risks of transmitting deadly neurological diseases. Recombinant technology solved all these problems overnight, giving us an infinite, completely safe, and allergy-free supply of medicine. With the safety issues solved, we now face an engineering challenge. How do we produce enough medicine for millions of people? The answer lies in the mathematics of bacterial growth. Bacteria do not just add one new cell at a time. They divide in half, doubling their entire population every single generation. When placed in a large, nutrient-rich tank called a bioreactor, this doubling leads to explosive exponential growth. By understanding and calculating this exponential growth, scientists can predict exactly how long it will take to grow enough bacteria to harvest a specific, profitable batch of medicine. This biological reality leads us directly to the mathematical model that governs it. The formula you see here calculates exponential bacterial growth. Let us break down what each part means. N sub t is the final number of bacteria we end up with. N sub 0 is our starting workforce, the initial number of bacteria we put into the tank. The base number 2 represents the doubling nature of bacterial division. In the exponent, T is the total time we let the bioreactor run, and G is the generation time, which is how long it takes for one cell to split into two. If we increase the total time, T, the exponent grows, and the final population explodes. If we decrease the generation time, G, meaning the bacteria divide faster, the population grows even more rapidly. Before we move on, let us see this formula in action with a concrete example. Suppose we inoculate a bioreactor with an initial population of 1,000 genetically modified E. coli cells. These bacteria have a generation time of 0.5 hours, and we let them grow for a total time of 5 hours. We want to find the final population. First, we calculate the number of generations by dividing the total time of 5 hours by the generation time of 0.5 hours, giving us exactly 10 doubling cycles. Next, we plug our starting population of 1,000 and our 10 generations into the formula. 2 to the power of 10 is 1,024. Multiplying this by our initial 1,000 cells gives us a final answer of 1,024,000 bacteria. Physically, this means our tiny starting workforce has multiplied into over 1 million microscopic factories in just 5 hours. Now that we have used this formula to understand how the factories grow, let us look at how they actually manufacture a specific medicine: insulin. Insulin is a crucial hormone that acts like a key, unlocking our cells so they can absorb sugar from the blood. Diabetics lack this key. However, making human insulin in bacteria is tricky. The human insulin protein is made of 2 separate short chains, called chain A and chain B, linked together. The problem is that bacteria do not have the cellular machinery to process and link these 2 chains the way human cells do. If we just put the whole insulin gene into a bacterium, it would not work. Therefore, scientists had to devise a clever workaround by synthesizing the DNA for chain A and chain B completely separately. This brings us to the brilliant solution scientists developed to bypass the limitations of bacterial processing. Instead of 1 batch, they use 2. They insert the chain A gene into 1 batch of E. coli and the chain B gene into a completely different batch. These 2 batches are grown in massive, separate bioreactors, where they independently manufacture large amounts of chain A and chain B proteins. Once the bacteria have done their job, scientists break open the cells, extract the individual chains, and purify them. Finally, in a controlled chemical laboratory, they mix chain A and chain B together. Under the right conditions, the chains bond naturally via disulfide bridges. This creates perfectly functional human-identical insulin, famously known as humulin. Let us visualize this entire process to make it perfectly clear. On the screen, you can see the dual pathway schematic for recombinant insulin. On the left side, notice the blue plasmid carrying the chain A gene entering its specific E. coli host, which then multiplies in its own bioreactor flask. Simultaneously, on the right side, the red plasmid carrying the chain B gene enters a separate E. coli host and multiplies in a different flask. The arrows pointing down show the extraction phase, where the individual blue and red protein chains are isolated. Finally, at the bottom, you see the crucial chemical step. The blue chain A and red chain B are brought together, and those vertical lines labeled S-S represent the disulfide bridges forming. This final structure is the active human insulin ready for patients. Having mastered insulin, let us turn our attention to another incredible success story: human growth hormone, or somatotropin. Why is this hormone so important? It is the vital signal that tells a child's body to grow. Without it, conditions like pituitary dwarfism occur. For decades, the only treatment was extracting the hormone from the pituitary glands of deceased humans. This was not only incredibly scarce, but dangerously risky, as it sometimes transmitted fatal neurological diseases. Recombinant DNA stepped in to save the day. Unlike insulin, human growth hormone is a single, long polypeptide chain. This structural difference is crucial because it means bacteria can read the DNA and manufacture the complete, fully functional hormone directly inside their cells, without needing the complex 2-batch mixing process we saw with insulin. Because growth hormone is a single chain, the engineering process is slightly different and quite elegant. Scientists start by isolating the messenger RNA for human growth hormone directly from pituitary cells. They then use a special enzyme called reverse transcriptase. This enzyme reads the RNA and converts it backward into complementary DNA, or cDNA. This step is vital because cDNA contains the exact streamlined instructions for the hormone, completely free of any unnecessary genetic junk that bacteria cannot process. This clean cDNA is then inserted into a plasmid vector and transformed into E. coli. The bacteria read the instructions, produce the hormone, and scientists simply break the bacteria open to harvest pure, safe recombinant somatotropin. Now that we know how the hormone is made, we must ask an industrial question. How much actual medicine can we harvest from our bacterial factories? This brings us to the protein yield formula. The formula is Y equals N sub T times M sub P. Conceptually, it is very straightforward. Y is the total yield of the recombinant protein we want to harvest, measured in grams. N sub T is the total number of bacterial cells currently living in the bioreactor. Finally, M sub P represents the tiny mass of the target protein that one single engineered cell can produce. If the total number of cells increases, our total yield increases proportionally. Similarly, if scientists optimize the genetic vector so that each individual cell produces more protein, meaning M sub P increases, the total yield will also increase significantly. Let us apply this yield formula to a real-world scenario. Suppose a pharmaceutical company has grown a massive population of 5 trillion cells, which is 5 times 10 to the power of 12. Each bacterium produces 2 times 10 to the power of negative 15 grams of human growth hormone. We need to find the total yield. We substitute these values into our equation. To solve this mathematically, we handle the numbers in 2 parts. First, we multiply the base numbers together. 5 times 2 equals 10. Next, we handle the massive difference in scale by adding the exponents of the powers of 10. 12 plus negative 15 gives us negative 3. Combining these, we get 10 times 10 to the power of negative 3. This gives a final answer of 0.01 grams. Physically, this equals 10 milligrams of pure hormone, which provides thousands of life-saving microgram doses from just one bioreactor run. We have covered a tremendous amount of ground today, so let us summarize the most important ideas. First, recombinant DNA technology revolutionized medicine by turning simple bacteria into living factories capable of reading human genetic blueprints. Second, by harnessing the power of exponential bacterial growth, we can scale up production to meet global demands rapidly. Third, we saw how different proteins require different engineering strategies. Insulin requires a clever 2-batch process to link separate chains, while human growth hormone utilizes complementary DNA for direct single-chain production. Ultimately, this incredible biotechnology has replaced dangerous, scarce animal and cadaver extracts with an infinite, safe, and highly pure supply of life-saving therapeutics. You now understand the fundamental biology and mathematics behind modern biomanufacturing.